Okay, good morning, everybody. And so hopefully in the next 25 minutes or so, I will shed some light on metal uptake, distribution, and storage in plants. Not surprisingly, this is a synchrotron image of an Arabidopsis thaliana leaf, and that's the model organism that I've done most of my work on. We've also done some work with rice. And so just shown here in green is potassium. We get a beautiful view of the leaf. Magenta is iron, and the blue is calcium. So I think most of you know humans require 17 minerals and 13 vitamins. And unlike vitamins, which are synthesized by the plants themselves, plants have to take up essential minerals from the soil. So that's really where food begins, and I know there are soil scientists here today. As Olena mentioned, iron is my favorite mineral nutrient, and we've already heard a lot about iron already this morning. It's the most common human nutritional disorder in the world. The reason for that is most people are getting their iron from plants. Plants aren't a good source of iron, although we'd like to fix that. And the reason plants aren't a good source of iron is iron's one of the 3 nutrients that most commonly limit plant growth. And this is a variation of the map Nigel just showed you with iron deficiency anemia. The darker the color, the worse the problem. And it's especially a problem for pregnant women and children. So Harvest Plus and other groups like the Bill and Melinda Gates Foundation are very interested in breeding crops for better nutrition. So iron uptake is highly regulated, as too much iron is toxic, and you can sort of divide all the plants in the world into 2 strategies. So the model plant that we work on, Arabidopsis, when it's starved for iron, it induces a series of processes. It's going to release small molecular weight compounds into the soil to help solubilize the iron that's there. And the iron 3 is the predominant form in most soils. They're also going to pump protons out to lower the pH. For every 1 unit drop in pH, iron becomes 1,000-fold more soluble. And then they're going to take this solubilized iron 3 and reduce it with a membrane-bound reductase that we identified in the lab a number of years ago called FRO2. And this is essential because it's the iron 2 form that's brought into the plant via the VIT1 transporter, also identified in my lab a number of years ago. And then the grasses, like rice and maize, they use a different strategy, more like bacteria and fungi. They release small molecular weight compounds to bind iron 3, and they actually bring the iron 3 complexes back into the root. So I'm going to tell you just 3 very short stories today. The first one I'll call localization of metals in the embryo. It's been worked on by a number of people in my lab over the years. So I just told you how plants take up iron, but of course we're interested in the parts of the plants that people eat, and a lot of what's eaten is stable foods are seeds. So they're a major food source for most of the world, but we still do not understand how important nutrients are delivered to and stored in seeds. And it's this very consumption of seeds with low micronutrient content that's causing all this micronutrient deficiency. So nutrient-rich seed would lead to agronomic benefits such as increased seedling vigor, resistance to disease and other stresses, and then ultimately would also increase crop yields. So we asked a very simple question in the lab, which is, where do seeds store iron? And our hypothesis was that plants are going to store iron in the vacuole, just like yeast. And then we had a second hypothesis, which is if a plant couldn't transport iron into the vacuole, then it would have less iron in the seed. And I just have a little diagram of a plant, but for those of you who aren't plant scientists, a vacuole is a large compartment in many plant cells that can be used for storage, and if you put metals in there, they don't participate in redox chemistry. So it turned out we were right about the first one. Plants do store iron in the vacuole. But we were wrong about the second one, because first we identified the vacuolar iron transporter that puts the iron in the vacuole, and then we had a mutant that no longer expressed the transporter. But that mutant still had the same amount of iron in the seed. And that's when Tracy Punchin came to my lab as a postdoc, and she was already a highly skilled user of the synchrotron, and she said, let's look and see where the iron is. And so that's really the entree for my group into the world of synchrotron science. So here is an Arabidopsis seed. I'm showing you a cross-section through the middle. And these were the very first images that Tracy generated at NSLS. And this is a wild-type seed. When we first saw the picture, we called it the 7 blue donuts. And then this is the mutant. This was the very first mutant we ever looked at, and it had a very different distribution of iron. So we knew we were onto something, because as I told you, each of these seeds had exactly the same amount of iron, 57 parts per million. So these blue donuts turn out to be a layer of cells that surround the circulatory system of the plant, the xylem and the phloem, and that's going to be moving iron from the root to the shoot, and also things from the shoot back down to the root. Now, does it matter if you mislocalize your iron? Well, of course, the answer is yes. So here's Arabidopsis and our vacuolar transport mutant growing in normally a little acidic soil that Arabidopsis prefers. They look fine. If we raise the pH to make iron less soluble, now you can see wild type's unhappy. It looks chlorotic, but it's going to do okay. Whereas the VIT1 mutant simply germinates and dies. So that was very exciting for us, and we identified a very basic aspect of the biology of a seed, which is you got to put the iron in the right place. And that's because this is the source for that germinating seedling before the root emerges and takes over. Now, I put this image in. This was taken at APS to show that not only have we gotten better at doing synchrotron work, but the synchrotrons have gotten a lot better. So now you can actually count the cells. There's 8 cells, and those are the cells that comprise the endodermis of the plant and surround the stele for the transport of nutrients. And then I'll also draw your attention to magenta. There's a single layer of cells, and these are the embryonic cotyledons that are inside of the seed, and they're accumulating manganese. And so when those seeds open, the manganese would be in a layer of cells, one layer in from the bottom surface of the leaf. We don't know why it's there. If anyone has any idea, that I should have put that on my list of unanswered questions. So anyways, it took us a number of years, like over 10 years, to finally get around to looking at what determines where the manganese goes. And to make a long story short, we did an educated guess. There's a family of transport proteins called metal transport proteins, and there were 4 of them in Arabidopsis that were thought to likely be manganese transporters. So what we did is we simply got mutants of each of those 4, 8, 9, 10, and 11. And then we went to NSLS and did a quick 2D scan. So now here's the root, the embryonic root. Here are the embryonic cotyledons. You can see in magenta the stripes of manganese, and the blue is iron. And so you can see down here in these 3 mutants, there's no change in the distribution of the manganese. But in the MTP8 mutant, now the manganese is co-localizing with the iron. So having this information from these quick scans, we then went to APS to look in more detail. So now here's what I was showing you again. It's a cross-section through an Arabidopsis seed. And here in wild type are those stripes of cells storing manganese. And these don't change in the VIT1 mutant, the iron transporter mutant. But now you can see in the manganese transporter mutant, now we're storing manganese in the same cells where iron is normally stored in a wild type seed. And then this is just showing you iron, where of course the VIT1 pattern looks a lot like the manganese pattern. Okay, so big question then, what happens if you get rid of both? And if you get rid of both of those transporters, you have no patterning in the seed. So it turns out that both VIT1 and MTP8 can transport iron and manganese. And what sets up the patterns is where those genes are being expressed. And so when one isn't there, the other one can transport the other element. So that was sort of satisfying in the end because there's only 2 genes required to set up the pattern of storage. And then I put this in, not knowing whether or not Nigel would be talking about this aspect, but when their paper came out, I was very proud of this work because they ended up using 2 genes identified in our group. So we're sort of the gene discovery end of the process, and you heard how long a process it is to bring something found in the lab out into the field. So as we already heard from Nigel, cassava overexpressing the iron transporter, VIT1 and ferritin, is the one they're moving forward. They had also looked at overexpressing the vacuolar transporter from Arabidopsis that I just talked to you about. So sometimes when I show this, and I'm not an author, people read the author list, but I get thanked for performing an advisory role. So anyways, I'm really impressed with what they've done. Okay, the second story is short. It's sort of our newest work right now, and that's localizing iron in the endosperm. So this is a project that was started by Seckin when he was a Fulbright scholar in my lab. He's now has his own lab as assistant professor in Turkey at the Middle East Technical University, and his student Dion is carrying out a lot of the work. So I'm just going to tell you how we got started on this. This is one of the original images from our paper on the vacuolar iron transporter, and so this is a 3D rendering. You can see the embryonic root, you can see the 2 cotyledons, and there was always this sort of blob of iron down here that we were ignoring. It's in the seed, but it is not part of the embryo. And I'm showing you the embryo here to orient you. So again, here's the root, and here are the embryonic cotyledons. And this region that we think is showing up here is the callosal endosperm. So the endosperm in seeds like Arabidopsis is a single layer of cells at the end in the mature seed, except for these 2 regions, the callosal endosperm. This is where the mother plant is attached to the seed, and that's where nutrients are going to enter. And then there's this region called the micropilar endosperm, and that's going to be important for germination. So we decided that maybe we should be interested in that little glob of iron. And I went back and looked at some old unpublished data that was collected by a student of mine, Maria Hint, with help from a great deal of help from Martin de Jong at the Australian synchrotron. So again, here now iron's in green, there's our radical, there's our 2 cotyledons. Here's that little cap of iron I was just talking about. And if we rotate the image and look at it top down, now you can see that this really is a defined structure. So, Arabidopsis seed are really tiny. They're the size of a poppy seed. So Seckin decided maybe he should go to tomato, where he could have a more substantial seed to work with. So this is an image, again, from NSLS2 that Seckin took. And so in tomato, the cotyledons sort of curl up at the end. But here's the callosal endosperm here. And I hope you can see there's just this tiny bit of iron here. That's the micropilar endosperm, we believe. And during germination, the endosperm cell walls have to weaken to allow that root to protrude, okay? So the seed coat has to rupture, but the endosperm also has to rupture. And this weakening was shown to occur through hydroxyl radicals that are generated locally. Usually NADH oxidases and peroxidases have been implicated. But Seckin's idea was because we have iron right here, close to where the radical is going to protrude, maybe this iron is participating in the generation of hydroxyl radicals via the Fenton reaction. And so that's sort of where his lab is going to go with this project with some help from us. They've done a lot of experiments already. They don't have time to show you, but If you add iron, germination is faster. If you chelate iron, germination is slower. And this trait, this ability to have all your seeds germinate synchronously is really important in agriculture. You can see fields yourself when you go, there's patches where, the plants are smaller, and so that's not good. So we think this, again, might turn out to be something of use once we figure it out. And here is a seed coat. So now the seedling has germinated. It's about 7 days old, the tomato. This is just the little seed coat that's left. And this is actually stained with a stain called pearl stain. But again, you can see this, there's still iron there in those endosperm cells that are left attached to the seed coat. So it's not really being used for nutrition. We think it might be used for fenton chemistry. Okay, and then my third little story I call Abundance, and I also like to say it's a 3 graduate student project. So it was started by Maria, she passed it to Garo passed it to Nabila, and Nabila is graduating in 2 weeks. Okay, so originally we weren't looking at where the iron was, we were simply measuring it by ICP-MS. And we were screening lots and lots of materials to look for ones that might accumulate iron. And after 100,000 samples, which is a lot of samples, we found an EMS mutant of Arabidopsis that accumulated ridiculously high levels of iron. So we were screening shoot material. So here, for example, a normal shoot in Arabidopsis might have 100 parts per million. These plants could accumulate upwards of 300 parts per million. They were also accumulating it in the seed and in the root. So maybe our job was over because it's already accumulating ridiculous amounts of iron in the seed. But of course, it's killing itself by doing so. So here's some synchrotron imaging that Maria did. And this just to drive home, here we have the mutants accumulating high levels in the root, the shoot, and the seed. We've also done some embedded work to look and see really more closely which cells. And these are cells that are near and dear to Olena's heart. Those are phloem cells that are accumulating large amounts of iron in the mutant. And I've labeled what the mutant's name is. It's brutus. Brutus was first identified by Terry Long, who had a Popeye and a Brutus mutant, so we got stuck with that now. And Can of Spinach just got published. Okay, from Elspeth's group. But here is the Brutus mutant. It's very unhappy in normal soil. It's taking up too much iron. If we make it alkaline, oh, now the mutant is really happy. And if we add iron back to the alkaline soil to prove it really is the lack of iron, wild type is looking great, and Brutus dies. Okay, so what is Brutus? What is Brutus? It's not moving. Okay, we're stuck. Oh, okay. It's just being slow. All right, so basically this mutant has a constitutive iron deficiency response, and that's because it has a mutation in this gene called Brutus. Brutus encodes an E3 ligase. It has 3 hemerythrin domains that can bind iron directly, and then it has this E3 ring ligase that's going to be involved in ubiquitination and turnover of whatever partner Brutus finds. So it is believed to be the iron sensor. It can bind iron, and then the cells know that they should proceed to turn over transcription factors involved in turning on the iron uptake response. So Brutus belongs to a small gene family. And I put this in, I'm not going to go over. We've been spending a lot of time on the regulatory cascade, but here's Brutus down here as a barrel attached to an iron molecule, and it's going to ubiquitinate and turn over these transcription factors, which in a cascade eventually turn on all of the iron uptake genes like IRT1 and FRO2. Okay, so we wanted to ask what happens if we look at mutants in this small gene family. There were 3 Brutus-like genes. So here's Terry's original allele. Actually, was a little less severe than ours, so we decided to use it when we were looking at the triple. And again, here's some synchrotron imaging, so you can see when we get rid of brutus-like 1 or brutus-like 2, there's not much change. And these are plants grown under iron deficiency. And here's the brutus 1 accumulating more iron. But look what happens when we make the double and then the triple. So this triple mutant, which is relieved of all this negative regulation, is now taking up a lot of iron, but in a good way, in that it's not killing itself under iron deficiency. It's looking, again, really nice, and it almost looks like there's too much regulation. The plant is going to great lengths to prevent itself from taking up too much iron. So we have good levels of chlorophyll, and the plants are doing well. So we decided to go ahead and start with the brutus mutant, which basically kills itself by taking up too much iron, and do a genetic screen. So we did the screen. So here's brutus, not looking great. We add the extra iron. All those little brutus plants will die, and we look for a mutation. We've mutagenized the line that's now able to grow. Okay, so we're just looking for survival, and we found one of the transcription factors that brutus is supposed to turn over. So if you get rid of this transcription factor called ILR3, now brutus goes from looking very unhappy to looking just like wild type. And then we go in and identify the mutation. As I said, it's a transcription factor, which made sense because we already knew it was involved in iron uptake. But what was unusual is when we looked under minus iron, here's, wild type goes chlorotic, brutus looks great. This mutant has these white sectors of bleached, dead, dying cells. So it has a very severe phenotype under iron deficiency. We now have figured out what that phenotype is, and that's oxidative damage due to singlet oxygen. And I won't show you all the gory details, but we're now in the thylakoids of the chloroplast. They're not unstacking, photo damage is occurring, and this is the transcription factor that's responsible for making sure that process gets underway. This image is the one that was actually in the paper. It's not so easy to see up here in the images. This is potassium, calcium, and iron. We were interested in what was going on in the bleach sectors of these leaves. And so down here, I've just done arbitrary fluorescence units across the arrow. So we're going from the bottom of the arrow to the top. And what you can see is when we're in a green area, we have relatively high levels of potassium, low levels of calcium. But then when we get into a bleached area, now we have low levels of potassium, high levels of calcium. And what we think is happening there is we have the cells are damaged, so we're getting leakage of potassium, and we can measure that, and we have. We don't understand why the calcium is accumulating in those areas of dead and dying cells, so that's something we're interested in. But again, it was using the synchrotron to tell us something about, the biology of what's happening. And cell death has been well studied. Oh, I forgot I put this in. This is a much easier one to see. So you can see, we're going from an area that has potassium to an area that doesn't. And then here's the accumulation of calcium in a bleached area. Okay, so we also looked at some other mutants. This turns out to be another transcription factor that's important for this photo protection. So again, you can see the depletion of potassium and the accumulation of calcium. And then just to drive home the story, this is a well-studied cell death mutant. And again, the same thing. We see the depletion of potassium and accumulation of calcium. So I'm going to leave you with a lot of unanswered questions, not all of which will be answered by our lab. Other labs are working on these too. When are the patterns set up in the seed? Do the patterns change throughout development? And then are these universal patterns, or do different plant lineages arrive at different solutions for metal storage? And here's just one unpublished example. This is a very young embryo, sliced through a young embryo. And zinc is in red, calcium's in blue. And then here's the mature seed, and you can see a very different distribution now of those ions. And I'm just a teaser. This is what my lab has been working on. We're looking at response to light, not synchrotron light, which is 1,000,000 times brighter than the sun, but sunlight. And after all these years, we discovered basically that our major iron uptake genes don't come on in the dark. So the plant has to be iron deficient and seeing light. So that's what I'm showing here. This is just some seedlings expressing reporter genes that have a luciferase. And so they shine in the presence of a substrate. You get light production. And so it turns out that we only see that in the light and not in the dark. And this little picture disappeared, but it's the exact same thing. Okay, so we're spending a lot of time figuring out how this all gets integrated, but it makes total sense that plants wouldn't really want to be doing a lot in the dark, except breaking down the starch that they made during the day. So with that, I'll just thank everybody in my lab that's worked on all these projects over the years. Lots of collaborators, funding from NSF, NIH, DOE, NIHS, Superfund, and I will just emphasize that almost all the people pictured have left my lab, and there went the synchrotron knowledge. So I have one remaining person that's really good on the synchrotron. So yes, that's an ongoing problem in my group. Thank you for your attention.